So next, it's really my honor to introduce our next speaker, Dr. Mooney, who is Director of Center for Information Technology at NIH. And he's going to talk about AI at NIH, drug discovery, clinical healthcare, and multimodal AI.
Good morning, everyone. Thank you so much for the opportunity to present today. I'm really, really excited to tell you a little bit about some of the work that I've been doing and others at the NIH over the 7 months that I've been at the Center for Information Technology. You might remember previous to this, I was at the University of Washington as a professor there, and I decided to take the big leap and go to the NIH. And it's been an incredible road so far to get here to where we are today. What I'm going to do over the next 25 minutes, I've got way too many slides, so hopefully I might have to skip some things, but I'm going to tell you a little bit about some of the work that the NIH is doing around investments in AI and kind of what our portfolio looks like, and then I'll talk about drug discovery and some projects that I think are really interesting that kind of help give an illustration of the kinds of work that both we're doing internally and well as in the extramural program. So first of all, I want to thank Susan Gregorek, who's not here today. She was originally slated, I think, invited to give this talk. I'm sort of giving this for her, and so I'll give a mixture of slides that are from me and from my office as well as from her office. If you notice, I'll put the logos of the various institutes and centers at the NIH on the different slides. It'll sort of give you an idea of where some of these slides are actually coming from. The NIH is 27 institutes and centers and an office of the director. CIT, which I've taken on, is one of them. We don't have an extramural funding program, so I don't actually, fund grants. I help support, IT and informatics and some other things that we can talk about, either now or during, either after the presentation or during a break or something. But we support a lot of the kind of things that we do, and I think you'll probably get a little bit of an impression on that. But I really want to thank Susan for a number of the materials here, and I know that someone from Susan's office is here, and I know there's some other NIHers here as well, like Julie from the NCI. All right, so first of all, I want to talk a little bit about my role, because my role is relatively new. I wear 2 hats. I have an NIH Associate Director hat. In the Office of the Director, I lead the Office of the Chief Information Officer, as well as our Cybersecurity Office. And I also lead the Center for Information Technology. We're about 1,300 or so people, and we support both enterprise computing for the NIH, but also support a number of the touch points in the extramural community, including our Cloud Strides program. If you have a Commons ID, we manage your Commons ID and things like that. And so really, we're trying to kind of think. I'm the first, I should say also, I'm a scientist. My predecessor was not a scientist. And I'm here to kind of help bridge the mission of the NIH with our computing that we do. And my hope is that at the end of the day, we'll have better tools that are maybe more efficient for managing data resources all the way through to the applications of AI. But it's not just me. Just like a school of medicine, that's one of the things that I've learned coming from a school of medicine. The NIH is 27 different institutes and centers and a number of different offices. All of them have their own CIO. We have 27 CIOs, I think, at the NIH, or more than 20 for sure. We all have data officers or data strategists, kind of leads within the different ICs and all, and all the different activities from them. And I think there's a very big challenge of coordination across all of that. I do want to shout out the partnership between the Office of Data Science Strategy, which I mentioned that Susan leads, as well as the National Library of Medicine. CIT and the Office of the CIO work together very, very closely on a number of challenges that support data science and the kinds of data science work that we do. So let me start by just kind of highlighting some of the broad, maybe not necessarily in the drug discovery space, activities that we do at the NIH. We have a vision for responsible AI and biomedical research. We lack the data resources necessary to optimally use AI methods to improve health and eliminate disparities. So there's a large effort to collect better data and AI-ready data sets. And we want to respectfully engage the people to earn trust, particularly when dealing with, like, say, real-world data, like healthcare data, or participation in clinical research. And we believe that data diversity is absolutely critical to the work that we're going to do, that we all do together in clinical research, to ensure that the outcomes of research, including AI, are equitable across all populations. And we need a dramatic increase in the collection of clinical care environment real-world data, including all populations. And you'll see some of those themes coming up. In FY23, we spent about $300 million for our AI programs officially. It's probably significantly more than that. In aggregate, it's more than almost $1 billion in research. I don't have the 2024 numbers yet to show you, but in general, there have been significant advancements in terms of the work that we're doing in AI to help and to help support that. And again, this doesn't include everything. Very interested in multimodal AI. This is very critical for drug discovery. You'll see some of that in some of my later slides. We're collecting significant amounts of data of different types of data. We've heard about omics data. We've heard about QSAR and traditional small molecule drug discovery. We've heard about real-world data, and I think all of these are playing together. And as we think about how those data might be integrated and brought together to enable for example, efforts in drug discovery or understanding healthcare and the outcomes of healthcare, I think that we have a powerful platform to enable research in the future. You may have heard of the Bridge to AI program, which is a program aimed at developing AI-ready data sets. Recently-ish, there were 4 projects that were funded generating Grand Challenge data generation. And the goals of the program are to, as I said, to generate AI-ready data, to develop best practices and ethical principles around these data sets and sharing of the data sets and models, and also getting exposure to help train and make the AI data scientists of the world more fluent. The 4 Grand Challenge projects that were funded were clinical care, an ICU-based project, a precision public health project, which uses voice as a biomarker, like digital recordings of, like, say, clinical visits as a way of helping identify risks of disease or diagnoses, support for disease. Salutogenesis, which is a complex word that you can read about the body's natural ability to heal itself and how to support that, as well as in functional genomics. Also an interest in NIH AI assurance labs under the executive order on safe, secure, trustworthy development and use of AI applications. We want to assist research in evaluating and mitigating the potential risks of AI and advance guidelines, best practices, and standards for responsible AI. There are a number of use cases that have been being developed between now, the fall of 2024, and spring of 2025. And we expect initial outputs to enhance the performance and ethics assurance for exploratory use cases of AI, develop, iterate, and maintain a framework for AI assurance, and inform assurance capabilities as well. So I would also like to talk a little bit about how we can prepare the next generation of biomedical research teams for ethical AI. I think you'll see that that's a common theme around equity and use of AI and how AI will impact populations that it's applied to and how we might be able to study it better. We really want to enable researchers as co-innovators of new capabilities and attract a broader workforce and spectrum of expertise within the ethical AI domain. You may have also heard about the AIM Ahead project. This is a very large, ambitious project with a number of universities and research institutions that have been involved with this. Maybe many of you have been involved with this as well. The AIM Ahead project is Artificial Intelligence Machine Learning Consortium to advance health equity and researcher diversity, and a number of organizations have been participating in order to help address health disparities and inequities, and also helping expose individuals, trainees, institutions that may not have the base of AI that we heard that, for example, Boston does to AI. I don't have time to talk too much about AI, AIM Ahead. There were a number of slides that I moved out, but AIM Ahead is definitely a program to look at if you're not familiar with it. It's a very good way to get involved in AI and NIH research in AI. As you know, the data is now at the heart of the NIH mission. So we run in CIT the STRIDES program, which is our cloud partnership between a number of cloud providers. Within the STRIDES umbrella, for you as an extramural researcher, you can get access to discounts in the cloud. Today, the STRIDES has more than 330 million gigabytes of data from basically every type of data that you could possibly imagine, and we support almost 2,500 investigating teams to help support the work that they do through the STRIDES initiative. And STRIDES is more than just oftentimes we think about it as like credits or a discount. What we're starting to see is an emergence of broader impacts from STRIDES, universities and investigators collaborating together to develop services that can help support, data science across to others. So for example, we've introduced Cloud Lab. This has been rolled out to the entirety of the extramural community. If you are associated with a NIH-affiliated institution, you can go to our web forum, you can log in, you can get $500 of free credits within one of our cloud partner sites, and you can get access to trainings that were developed by Idea State CTRs collaboratively that build upon our STRIDES program to enable doing that. We all saw that AlphaFold and the ab initio structure prediction, David Baker won the Nobel Prize a week ago. This has definitely been the year of AI in the Nobel Prize world, and we know that this has been incredibly impactful in terms of all the work, the work that we do. I do want to shout out the NIGMS-funded CASP competition, the Critical Assessment of Structure Prediction. I think that, 30 years ago, 35 years ago, a number of investigators got together to say we need to have an unbiased assessment of how well structure prediction algorithms are working together. And without that unbiased structure, that unbiased assessment, we wouldn't know which methods were doing better than others. And we would have never known, in my opinion, that AlphaFold was able to have solved the problem, because obviously when we write papers about our own methods, we always say they're great and we always compare them in a way that peer review gets excited about them. And I think that I really want to emphasize that CASP is absolutely critical to this. And I think these sorts of benchmarking experiments where we work together to try to, in an unbiased way, assess the accuracy of methods to quantitative endpoints that we can measure as a gold standard is really critical to advance AI in the future. And there's a community of critical assessments out there. There's commercial like Kaggle, and there's some open data science ones like the DREAM challenges. I'm a huge proponent of these and have been a part of a lot of them in the past as part of my research activities. So obviously I'm a proponent of that. I think we know that there's tons of activity, tons of work that's been done around using, artificial neural networks to look at, predicting protein structures, but there's still an enormous amount of work to do. We heard about protein docking and use of AI to use of these kinds of machine learning models to predict, like, say, drug candidates to targets, but there's also things like structural changes that might happen in the presence of genetic variants that are within the human population that might affect protein structure or function. The dynamics of proteins also has something that's been very well studied that AI may also be able to contribute to. And so I think there's an enormous amount of work of looking at this predicted structurome of proteins to try to understand, how we might be able to translate into better things such as drug discovery. At the NIH, we have funded projects both internally in our intramural research program as well as extramurally to many of you probably that are focused on the spectrum of drug discovery work, everywhere from refining small molecules and looking at pharmacophores that might be useful for drug discovery, assisting in high-throughput screening, modeling and predicting protein structures, support for digital twins and creating basically a digital image of what a patient or a clinical trial participant might look like, generating organoids and lab-based tissue that can be used for screening, and disease models in a petri dish or in a high-throughput screening session, pharmacogenetics and understanding the impact of our human population of genetics affects our response to drugs, translating that to animal models and understanding how animal models are relevant to humans and how they can be translated back, and then using all of this data to think about drug repurposing. We've already heard a talk about that earlier, and then modeling clinical trial data, an area called pharmacometrics, which is very well understood in the pharmaceutical world, but maybe less understood in the academic research world. And then moving to humans and trying to understand clinical trials and improving the identification of participants within clinical trials using AI, as well as leveraging clinical data, improving clinical data, standardizing clinical data, getting access to clinical data, and also then leveraging clinical data better to develop better gold standards, better models that can predict things like, say, for decision support about whether a patient is at a risk for a disease or other things that we might see within a clinical care setting. I want to talk a little bit about some stories of some funded projects. So for example, Long Li, who I think is chair of biostatistics at Ohio State University, is funded from the National Library of Medicine to leverage AI to look at drug interactions and predictions of new pharmacogenetic effects, basically starting with all of the literature that we know in the world and leveraging AI methods to then identify potential drug interactions and then using them. So for example, he is using a well-known and well-leveraged tool called BioBERT. He is able to parse millions of abstracts that are present in PubMed and identify PK and PD effects that are drug interactions that are interacting to support PK and PD effects, can then hypothesize individual pairs, and then can look at these different pairs and whether they are affecting both PK and PD based on data that's available. Things that affect everything might be pharmacogenetic, might actually be a genetic cause, and then can go into patient data, like using the Indiana Network of Patient Care or the Adverse Event Reporting System from the FDA, and identify whether his hypotheses are in fact from those observations are in fact statistically interesting, and then can then build a model of how, for example, in this case, of how interacting drugs may be affecting a pharmacogenetics pipeline, like say a cytochrome P450, and then can actually go and look at a real world biobank clinical database like BioView, I think at Vanderbilt, and assess whether the hypothesis that's been built and the structural model seems to play out in real people from real data. We've also talked about drug repurposing. Another project that's funded by the National Institutes of General Medical Sciences and the National Institutes of Aging is Lee Shea's group at Hunter College and City University of New York. And he is basically leveraging all the kinds of data that I just described to apply a method that he calls AI QSP to essentially look for potential drugs that may be applicable in different diseases that are already approved, much like we heard about in the first keynote speaker. And so this approach of AI-powered quantitative systems pharmacology, I'm not going to talk too much about this, but he's leveraging our knowledge of small molecules from cheminformatics databases, things like the Links 1000 data set, which was originally developed just down the street, where we can take small molecules and see how they change gene expression on cell lines, to real-world data, to other things that are available out there, to then try to hypothesize potential data sets that might be, potential molecules, small molecules that may have effect. And he can apply those collaboratively with others in rodent models. In this case, it's a drug that he's looking at as a candidate that he's looking at for potential repurposing into Alzheimer's disease. And from those rodent models, he can then hypothesize mechanisms of how a certain molecule that seems to be affecting or ameliorating the rodent the rodent model, he can then take those and look for pathways that are being changed through gene expression within those rodents and collect other data to look at potential clinical outcomes. Within the NIH, the National Center for Advancing Translational Sciences, it's one of our institutes. They also fund the CTSA program, which you're probably familiar with. Internally with them, they have an incredibly impressive high-throughput screening and other technology platforms for screening large amounts of small molecules. And as you might expect, the integration of AI has become really critical in terms of the process of where in the old days we would take a library with basically these large garages of small molecules in little vials, like every pharmaceutical company probably had one, and you could automatically pull it out. Yeah, sure. And you could automatically pull them out and then run a screen in 96 well plates or something and look for hits. And nowadays, we can actually apply AI to do a whole wide range of the sorts of things that we would want to do with high throughput screening to help support that. I'm not going to talk too much about this, but just want to point out that internally within NCATS, and they're always looking, I think, for collaborators, potential collaborators, to help improve the high throughput screening process with AI. So for example, you can imagine applying AI-based QSAR based on previous high throughput screening experiments, data that's available, and building an AI method that can then build new potential candidate molecules based on the previous high throughput screens or other data that you train into it. But these molecules are also synthesizable. So that means that it's not just some random molecule that has a set of pharmacophores that you think might be interactive, but you also know that you can make it and that it's drug-like, and that these candidates can then be taken and then experimentally screened. And the AI working together with the experimental screen is coming up with small numbers of candidates that then could be then taken to other preclinical work, preclinical experimentation. It doesn't just end there. Another project that's also funded by GM as part of this kind of collaborative innovations or research project is looking at natural products research. There's a huge amount of heterogeneity in terms of the diversity of life on Earth. Many molecules are produced by these organisms that are bioactive that might be potential drugs that could be screened against cancer or other diseases that we might want to look at. The problem is they're hard to manufacture. They're hard to synthesize. Some of these molecules can be quite complicated, and they are hard to study because we generally only have very small amounts of them. And so Dr. Hendrik Lusch, who I've put here from the University of Florida, has developed a collaborative team where they're going and looking at cyanobacteria and sponges that they're pulling out, and they're applying AI through the whole process, sequencing genomes, identifying the BGC bio something clusters. It's basically the biogenic clusters. It's basically the genes tend to be next to each other that produce these metabolites or the small molecules within them. And so you can use AI to predict the set of genes that are going to produce those small molecules. You can clone them out and actually try to synthesize them and use them to screen against whatever it is that you want to look at. And AI is part of every single step in going from taking a sample from the ocean, identifying their genomes and what metabolites are being produced and what the genes are doing to produce them, and then using using cloned sets of those genes to then express them, and then you can do screening against something to whatever it is that you want to look at based on the molecules that you discover and predict. A couple of other things I'm just going to mention. I'm almost out of time here. Another very innovative R21 by Paul Peng is using very similar technologies from Greenstone Biosciences, is using very similar technologies to what I just described a second ago, but it's using iPSCs to generate, essentially like organoids for high throughput screening, doing AI very similar to what I just described. We're using ANNs to then predict, potential candidates or predict basically anything that we would want to predict, and then using organoids that have been developed to model specific diseases in a high throughput way that then those AI-based small molecules can be screened against. A couple of other things to mention that are not necessarily deep in drug discovery, but I think are really, really cool. Nick Tatanetti, who's at Cedars-Sinai, has been doing very, very interesting work in looking at extracting adverse drug events from drug labels using natural language processing. He has developed methods and a database. It's all open. You can get it from GitHub and yeah, thank you. I think I've got like 3 slides left. We're close. And you can access that and download that data of these are known extracted drug interactions from labeling into a single database, again, using AI, and very useful for pharmaceutical research. Finally, I want to shout out Nick Dobbins. He did this work when he actually worked for me at the University of Washington. He's now a professor at Johns Hopkins University. He developed something called Leaf AI, which is a AI-based natural language cohort discovery tool for real-world clinical data. So you can enter in, tell me how many, find patients with Parkinson's disease that were treated with some drug that were in an emergency room in the last 6 months. And it'll come back and tell you how many patients are in your enterprise data warehouse from your clinical site, meet all of those criteria. And he has shown that that algorithm outperforms a human database query by showing that the algorithm is better predicting of patients that actually enroll in clinical trials based on using clinical trial inclusion criteria as the input to this method. All right, tons of new opportunities. I'm over time, so I'm just going to kind of quickly go through these in AI of things that are actually happening to develop, new data, synthetic clinical data sets for AI to make AI easier to distribute, and new technologies, as well as broadening the kinds of partnerships that we do. So over the next 5 years, we will improve capabilities to sustain the NIH policy for data management and sharing. We'll develop programs to enhance human-derived data for research, new opportunities in software, computational methods, and AI, and support for federated biomedical research data infrastructure, particularly real-world data, connecting together patient data for research use and strengthening that community of individuals that we train. We're finalizing our next strategic plan for data science. It's available, you can read it, and it should be available, I think, finalized very, very soon. I don't know the timeline for that, but it's out there. And with that, I think the AI future is very, very bright, and I want to shout out that if you want to hear more about what we do in CIT, and the technology that we're going to be producing, you can follow me on LinkedIn. If I post it on LinkedIn, it's important, I promise. And that's and we're going to have a newsletter that'll also be posted there that'll talk about computational investments. And then, of course, Susan's team at ODSS has been transformative in really making data science work at the NIH. So with that, I want to thank you very much and the organizers for the opportunity to talk. And I think I'm about 2 minutes over, and I apologize to taking away time from others for that. Thank you.
Thank you. Thank you very much for a really informative presentation. I particularly want to thank you for calling out the fact that there would have been no AlphaFold without CASP. So that really points to the importance of open data and benchmarks. And by the way, that's one of the topic of one of our breakout sessions in the afternoon. So, those who are interested in this topic, make sure you participate.
Thank you very much.
Thank you again.